Hello, everyone, and welcome to today's presentation titled Identification of Novel Blood-Based Signatures of Neurodegenerative Disease Using High-Sensitivity NULISA Protein Assays. This webinar is part of the 13th annual event in the Neuroscience Virtual Event Series. I'm Israel Gonzalez, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and sponsored by Alamar Biosciences. For more information about Alamar Biosciences, visit alamarbio.com. Now, before we begin, I would like to remind everyone that this event is interactive, and we encourage you to first participate by communicating with other attendees using our live chat feature during the presentation. You can find the live chat located at the left of your screen. If you have any trouble seeing or hearing the presentation, you can click on the Help Desk button located at the top of your screen within the navigation bar or from the lobby within the event. Now let's get started. I would like to present today's speakers. Doug Hinerfeld, Senior Director of Application Support at Alamar Biosciences. Maura Malpetti, UK DRI Emerging Leader Race Against Dementia, ARUK Fellow Senior Research Associate at University of Cambridge. And last but not least, Sterling Johnson, Professor of Geriatrics and Dementia, Department of Geriatrics and Gerontology at University of Wisconsin. For a complete biography of our speakers today, you can visit the Presenter tab from the menu on the left of your screen. Doug, Maura, and Sterling, you may now begin your presentation.
Hello. Today we're going to talk about the identification of novel blood-based signatures of neurodegenerative disease using the high-sensitivity NULISA protein assay. As an introduction to myself, my name is Doug Hinerfeld. I'm the Senior Director of Field Application Support here at Alamar Biosciences. I'm going to talk a little bit about the NULISA technology, and then I will turn it over to Dr. Malpetti and then Dr. Johnson from University of Cambridge and University of Wisconsin, and they're going to talk about the application of NULISA for the discovery of biomarkers for neurodegenerative diseases. So at Alamar, our mission is powering precision proteomics to enable the earliest detection of disease. How do we enable that? We focus, so it's a proteomic platform, and we focus on sensitivity, so femtogram per mil sensitivity for the proteins in our panel, having an extremely large dynamic range, 12 logs, so that you can look at low abundant proteins and high abundant proteins without performing multiple dilutions of your sample. Multiplexed analysis, being able to look at 250 or even more plex numbers of proteins, and a fully automated platform. So it's less than 30 minutes of hands-on time, but I think the other key point about automation is it really reduces the variability in the data. So the NULISA platform, as I described, it's really best-in-class attomolar level sensitivity to increase the depth of the measurable proteins that you can detect in biological fluids, serum, plasma, CSF. You can detect critical disease biomarkers at even lower levels, discover novel biomarkers and drug targets that are currently missed by other platforms that aren't as sensitive. And importantly, even in healthy populations, the sensitivity gives you the ability to detect the baseline expression of these various proteins. So there's sort of three components to the NULISA platform. There's the consumables themselves. So for each assay that you run, be it our CNS disease panel, which you'll hear more about today, our inflammation panel, or our other panels, all of the reagents are provided within a kit. There's the instrument itself. It's a very easy to use instrument. It's a touchscreen instrument. And you'll see on the right, there's 3 bays. Each bay can run an independent assay. So you could run the CNS disease panel in bay 1. You could run the inflammation in bay 2, and you could run one of our singleplex assays in bay 3. And then we have a suite of software, both for setting up your experiments, what assay you're going to run, what samples you're going to run, and for QC and analysis of the resulting data. So the workflow itself is incredibly simple. Once you've set up the experiment in our software to say, again, what assay, what samples do you want to run, you simply plate those samples into a provided microtiter plate. You load it into the instrument. For a single-plex assay, the onboard qPCR machine provides the quantitative results for that marker within 8 hours. For the multiplex assay, at the end of that run, you get a single well within our kit that has a sequencing library. That sequencing library is simply quantitated on a qubit, loaded on a sequencer, sequences in about 8 hours, depending on the platform, and then the resulting sequence data is processed by our software, ultimately to give you the expression levels of all the proteins across all of the samples that you ran in the assay. So the advantage of the platform is really this ability to go from sort of a focused discovery, focusing on hundreds of markers that are very focused on particular biologies of interest, down to having flexibility to focus in on proteins of interest. And then ultimately, the future is transitioning this into a diagnostic assay. So NULISA, which is at the heart of all of this, is an immunoassay. It uses a pair of antibodies specific to the antigen of interest that are conjugated to DNA oligos. And I'll talk a little bit more now about sort of how that works and the benefits of the platform. So it starts, you've loaded the samples, the antibodies are added, and this all again happens within an automated system. The immunocomplexes are formed. We then use magnetic beads, and we first bind an oligo-T bead to the poly A tail, wash away everything that doesn't bind. We then release that complex as well as the free polyadenylated antibodies. We then bind to the other side to a magnetic streptavidin-coated bead binding to the biotin. Again, perform a wash. Now at this point, this dual capture and washing has removed all the background, and that's what really gives us the increase in sensitivity and therefore the increase in dynamic range. At this point, we add this bridging oligo, we do a ligation, and then if you're running the singleplex, it goes onto the onboard qPCR machine, and we simply quantitate the presence of that specific ligated product. If we're going into sequencing, where we have a whole mix of products, we then sequence those, and again, that data is sort of deconvoluted such that you can analyze what is the expression of each protein in the panel in each sample that you loaded on the microtiter plate. With that increased sensitivity, if we compare it to just proximity ligation assay alone on the left, we see this dramatic increase in sensitivity and dynamic range. And then on the right, using the same antibody pair and comparing directly to the Simoa platform, we again see an increased sensitivity and noticeably an increase in dynamic range of the assay versus Simoa. So this kind of sensitivity, attomolar or femtogram per mil level sensitivity, we consistently see that across many different proteins, both in the singleplex assay and in the multiplexed assay. So just an example of that is our IL-1 beta assay here, showing a comparison between NULISA and Simoa, showing improved sensitivity down to 3 femtograms per mil for NULISA. And just you can see the amazing performance of the assay. Again, this huge dynamic range that we're able to see, the very high performance in terms of parallelism and spike recovery. So that's all kind of looking at singleplex assays. And here what we've done is we've looked at the same proteins in the top row, either as singleplex assays or within the panel. So same antibody pairs, but added to a large panel of 250 different proteins. And what we can see is we get nearly identical results, whether you're looking at it singly or you're looking at it in the context of a panel. So you can transition very smoothly back and forth between singleplex assays and multiplex assays. With that level of sensitivity that we can get in the panel, we've now developed a whole series of different panels, and we will continue to grow our portfolio here. So this is the NULISA-seq inflammation panel, which has about 250 different proteins in it, cytokines and chemokines and growth factors and so forth. So importantly, it only takes 25 microliters. You load 25 microliters of sample into the microtiter plate. It actually uses 10 in the assay, but you load 25. We get consistent attomolar level sensitivity, or 10 femtogram per mil. Again, without any dilutions, we get 12 logs of dynamic range. And what that means is that you can detect proteins, down at attomolar level and, way up to much higher abundances, all without any dilution of the sample. And with the automation, it's highly reproducible. So looking at the same samples across multiple wells, multiple runs, multiple instruments, we consistently see a CV of less than 10%. And again, this highlights the content within our inflammation panel itself. We also have a version of that panel that provides absolute quantitation. So here you're getting actually picogram per mil or femtogram per mil quantitative levels of the proteins within the panel. As you'll hear more about today, we have developed the CNS disease panel 120. You'll see, looking at the content in the upper left with the amyloid and tau pathologies, all the important phospho-tau markers and A-beta markers, also proteins involved with synuclein and synaptic function, TDP-43, alpha-synuclein, and so forth, vascular, neurodegenerative markers, and then a whole host of inflammation markers as well. We are now running in our technology access program, and soon we'll be launching our NULISA-seq mouse panel 120. Again, great content for immunology and inflammation, neurobiology, again, looking at things like TDP-43 and phospho-TDP-43 and alpha-synuclein and phospho-taus and so forth, as well as a host of markers that are quite valuable and interesting in the context of oncological research. So, of course, you can build the best panels, but if you can't detect the proteins in the panel, it's not much use. This is, so each one of these little bars represents an individual protein within the panel, and then the distribution of expression sort of up and down over about 40 different healthy human samples that we ran. And what you can see is that 99.6% of all the targets in the panel were detectable in at least 50% of those samples. So, you can see even very low abundant proteins here, such as IL-1B and IL-17A, for example, detectable in normal human plasma. The advantage of this is, of course, if you're looking at large populations, you get real values for even the healthy population, not just sort of zero values, and then only seeing increases in expression. So the sensitivity has clearly huge value in that context. Something that is also coming in just really a few weeks now is our NULISA qPCR custom assay development kits. So what this is going to allow you to do is to create your own assays. So you'll be able to take antibodies that you've perhaps been using in other types of assays where maybe you haven't gotten enough sensitivity or dynamic range. We provide a kit where you can conjugate the oligonucleotides to it and then develop assays using our kits that are ultimately fully automated on the Argo instrument, really expanding the capabilities of the platform. So you can see a lot more detail about the assay and the instrument in our Nature Communications article. The barcode is shown here, so you're free to download that as well. And with that, I'm going to turn it over to Dr. Malpetti.
Thank you very much, Doug, for that brilliant introduction to the technology. And I'm very happy to be here today to present a bit of our experience with these panels and how we are using these for our research, in particular across different neurodegenerative diseases. So here are my disclosure. Oh, no, sorry. These are my disclosures. And here a bit of the background, as most of the audience here will know, has been quite a revolutionary of blood-based biomarkers in the last few years in the background of dementia. And in particular, most of the efforts have been in developing different markers for pTau or amyloid pathology, and more peripherally also neurodegeneration and neuroinflammation. There are some remaining challenges despite the excellent progress that we are doing. In particular, we are trying to focus on bringing these different markers into implementation in clinical practice. That is definitely our final goal. We need better markers for differential diagnosis, especially in non-Alzheimer and non-Alzheimer pathology. And also we need to identify a bit more the confounding factors like environmental or also the technology related factors that we need to be aware of when we collect this sample. But very importantly, also, we are trying to use these blood-based biomarkers to identify treatable pathways. So in this context, I think that larger proteomics panels are very important because they allowed us to explore beyond just the specific markers that we are interested for diagnosis and prognosis, but also to identify other pathways that could be treatable in the context of dementia. And in particular, we are interested in synaptic inflammation, vascular changes, and also other proteinopathies. So with this in mind, we decided to apply the NULISA technology, as Doug already introduced very well. I will not enter in the details, but definitely this was very appealing as a technology for us because it allowed us to apply a multi-analyte proteomics analysis, so looking at different markers at the same time at attomolar sensitivity and especially in low blood volume. And I think this is very important for future studies. There have been a few publications now out there, and we wanted to also join this effort in looking at these panels in different diseases. In particular, we applied both the NULISA CNS panel and the NULISA inflammation panels in 172 samples. And I highlight here that for our pilot study, we look at serum samples rather than plasma. And we look at patients with Alzheimer's disease or mild cognitive impairment that were positive at amyloid with amyloid markers, patients with Lewy body dementia, patients with frontotemporal dementia, and patients with progressive supranuclear palsy. And if you are interested to read a bit more about this study, it's been just accepted yesterday in Alzheimer's and dementia, and it's in press, so it will be public quite soon. So just to dive a bit into the results of the project, we started with some quality control because it is a real-world observational studies. What we are running in Cambridge, we have samples that have been stored in the freezers at -70 for many years, and so we wanted to double-check that the NULISA protein quantification was not affected by this storage time. And very importantly, we found that the target detectability was quite high for both panels, more than 94 and 97 respectively for CNS and inflammation, but also the storage time, very importantly, didn't have any impact on the quantification. Then we wanted, like in 92% of these participants, we had also plasma samples that have been analyzed with different markers, in particular pTau with the AlthPath assay, pTau 231 with in-house SIMO assay from the Gothenburg group, and also we applied the Quanterix SIMO human neurology 4-plex assay for amyloid, GFAP, and NFL markers. And as you can see from these graphs, the correlations between the quantification of NULISA in our serum samples and the quantification of these markers with in plasma with other technologies was quite high, and these in particular for pTau 217 and NFL, and this was replicated in previous studies as well. So in terms of CNS markers, we started with comparing groups, given that we have this very heterogeneous group of patients. And we saw that comparing controls versus patients, we identify pTau, in particular 231 and 217, but also 181, as primary markers distinguishing patients with Alzheimer and patients with dementia with Lewy body as compared to controls. And this is not surprising. surprising, but it's very important to see that in addition to this, actually, for more the explorative approach that NULISA can provide, we also identify some further markers for neurodegeneration, synaptic and inflammation markers, especially in the LBD group. Very interestingly, when we move to patients with PSP and frontotemporal dementia, the major marker was NfL, not surprisingly, and as a neurodegenerative marker, and the pTau were actually not the top hit of the panel. But very interesting, as you can see from these graphs, there was quite a lot of inflammation markers coming up, especially in the context of the patients with FTD. So then we moved to, rather than just distinguishing groups from like patients from controls, we wanted to see what was predictive of clinical progression and eventually survival. So we look at the top markers of interest like NfL, pTau 217, pTau 231, and GFAP as compared to age and sex and group and the diagnosis. And as you can see, in terms of survival, the most predictive marker across all these conditions was NfL, with a dramatic drop in terms of survival time with higher levels of neuroinflammation, of NfL levels in the serum of these participants. Very interestingly, also something that we were not looking out specifically for, but we identified with the panel, we identified one marker in particular that was higher in controls systematically as compared to all groups of patients. And this is the corticotropin releasing hormone. As you can see, the level were higher in controls as compared to all groups. And also this marker having lower levels of this marker was predictive of how fast these people decline and eventually die. So this is very interesting, and just to give you an example of like while you are looking for something in these big panels, you can also identify something else that is data driven and but still very important biologically. In Cambridge, we are also very interested in inflammation, so we wanted to go beyond the CNS panel, even if the CNS includes some of the inflammatory markers that we are interested in. And we wanted to expand a bit more to the 250 panel for inflammatory markers. So in terms of inflammation, just to give you a bit of a background, in case you are not familiar with this concept in dementia, so in neurodegenerative diseases, we have chronic inflammation that is definitely driven by cells that are resident in the brain, in particular macroglia and astrocytes, and this hyperactivation of these cells leads to various aspects of the disease, but also storm of cytokines and chemokines. And there is more and more evidence showing that actually there is a constant discussion and interaction between the peripheral system, immune system, and the central immune system. And we can capture that with some markers in the blood, including cytokines and chemokines. Looking at centrally, we wanted to, we use different markers, in particular PET imaging, and that is the validation, the gold standard that we are using to look at what happens in the brain. And we saw that in various diseases, we show that higher inflammation is actually associated with more rapid decline. And for that reason, we are much more interested in looking at what happens as a wall, as a system, like between peripheral and central, to maybe find something that can slow down these conditions. So as Doug introduced already, there is this big panel that covers different aspects, different markers, cytokines, chemokines, receptors, etc., and gives us the possibility of looking at the scalability of our previous work with imaging, going for something that is more accessible, and looking at multiple targets and have complementary information between central and periphery. So with in our samples, looking at patients versus controls, we identify actually quite a lot of markers, around 57 markers that were upregulated or a couple of them downregulated in patients as compared to controls. And as you can see, there are like some famous markers out there, like IL-1 beta is there, CSF1 is there, GFAP is there. So it's very interesting to see that actually some of the markers that we were expecting are coming up in this data-driven approach. Also, very interestingly, when we look at these, each disease group as compared to controls, we identify different patterns of inflammation, and we had like groups of around 35 patients per each condition. And as you can see in the patients with Alzheimer's disease and multi-cognitive impairment, the major driver of this inflammatory pattern was GFAP, which has been shown in many papers that as plasma markers actually is very good for Alzheimer's disease related associative reaction, and in particular related to amyloidosis. But in the PSP and NFTD, there is like a much broader panel that comes up with different markers and many more cytokines to explore that could be quite relevant for this specific condition as compared to, for example, Alzheimer. Very importantly, also in our groups, we look at the CRP as a marker which is part of the NULISA panels, and it was not elevated in any patients groups as compared to controls. And this is very important to exclude any non-specific systemic inflammation. So then we went for trying to understand how these inflammatory patterns can predict also survival. As I said, like with the PET imaging, we showed over and over that inflammation in the central system is related to much worse clinical decline over time. And we wanted to basically identify if there was something that we can capture also peripherally. Looking at the top markers across all groups, there was in particular this CD276 that was coming up as a marker that was predictive of faster faster decline and shorter survival. And this has been a marker that has not been looked much in dementia, but much more in cancer. So it was something that we were not looking necessarily for, and it's something that definitely we need more studies to understand their role into dementia and neurodegenerative diseases. And then we went for a more explorative approach, looking at all 57 markers that were identified as elevated in patients versus controls. And we applied a machine learning selection of the markers that were predictive of much shorter survival. And as you can see, the final model identified the markers that were relevant for this clinical outcome. And as you can see, like the top markers that were related with much worse outcome in terms of shorter survival was IL-1 beta, and in addition to also which diagnosis this participant had, but also IL-15, which has been described in several studies as well as IL-1 beta as implicated in different conditions related to dementia. And also there are other markers that you may recognize there. And very interestingly, also the S100A12 is something that has been recently published from another group showing that is quite relevant in Alzheimer's disease. And while if you go down and see on those markers that actually were related with longer survival, so in some way protective, we identified among others the TREM2, which is quite an interesting finding. This confirms our previous studies with other technologies that were less sensitive than NULISA, but so basically the NULISA inflammatory panel allowed us to look at a much larger marker, but also confirming that previous studies have been showing biologically relevant informations. And in particular, in our previous study, we look at a panel of cytokines, and we identified that, again, there is a pattern of inflammation that is elevating in patients with frontotemporal lobe degeneration as compared to controls. And this relates with central inflammation as measured by TSP PET and predicts survival. So the next step is definitely trying to do something similar with the NULISA panels to try to understand a bit more in those extra markers with much more sensitivity, how this relates with the central inflammation and with the clinical outcomes, and potentially identifying targets that could be helpful for switching off the harmful inflammatory patterns in these patients. And something that we are collaborating with the group of Amanda and Eric, UCL, is to expand our research not just from one center, but actually across different centers across the UK to validate basically these markers across many participants, especially with frontotemporal dementia, which is our main focus, but also trying to understand how different factors can influence the quantification of these proteins and the patterns, in particular looking at ethnicity, genetic and socioeconomic diversity, and other aspects. So this is a new study called On Fire that we just launched in the UK, and it will be very exciting to see how the NULISA platform can help us to answer some questions in this bigger study. So just to summarize, I show you that NULISA has high sensitivity and reliability, and and the quantification strongly correlate with CIMOA for pTau, 217, pTau, 231, NFL and GFAP. We identified that in patients with Alzheimer's disease and Lewy body dementia, the pTau 231 and 217 were the top hit to compare them as to controls. And in FTD and PSP cohorts, actually it was NFL. And this also was predictive of survival. I hope that came across also that this is a very good platform for discovery of new markers, and not just looking at the top 4 markers, but actually looking at patterns that are like more data-driven and involving other aspects of the disease, and in particular showing upregulation of inflammatory markers and different patterns that are associated with survival. With this, I wanted to thank you for the attention, and also all my lab in Cambridge, the collaborators, of course, amazing people working behind the scene, and all funders that made this possible, and Alamar for organizing this. Thank you so much.
Hello, I'm Sterling Johnson. I'm delighted to talk to you today about Alzheimer's disease and the insights that we are gaining from blood-based biomarkers and other biomarkers on this disease and other forms of neurodegeneration. This is work that has been done by a team of people, especially Rachel Wilson, Ramiro Reyes, and Julie Homans with me, and an expanded team around us. Let me start by conveying a little bit of the problem we have in our field. Right now, we are coming to grips with the fact that Alzheimer's disease does not occur by itself most of the time. In fact, it seems to co-occur with other forms of neurodegeneration. These things take decades to evolve. We do not know why they may co-occur, why some people have more than one etiology and others do not. These proteinopathies may be asynchronous in their onset ages. An Alzheimer's disease proteinopathy may begin at age 60, for example, and another proteinopathy in that same individual, say TDP43 or alpha-synuclein, may occur at a later age. So we need tools to understand these asynchronous onsets and differing rates of progression, and just the presence of these multiple pathologies. Well, this is the approach that we are trying to take, and we're using blood-based biomarkers to help us do that, because right now these tools don't exist, and we need to be able to get there. And this is here on this slide an illustration of the problem. This is a Venn diagram of the overlap of Alzheimer's disease neuropathologic change, ADNC, with TDP-43 proteinopathy and Lewy body or alpha-synuclein proteinopathy. And what we see here is that when AD neuropathology is present, about 65% of the time, at least one of those other two proteinopathies is also present in the brain. And this is new data in 2024 from Woodworth et al. Published in Acta Neuropathologica. So we have been looking at the neuropathology literature and listening to our neuropathology colleagues who keep telling us that Alzheimer's disease does not occur in a vacuum. And in 2023, we put together a proposal that was funded by NIH to bring together all the Alzheimer's disease research centers to try to get a better handle on the different etiologies that may be present in our patients. And so that is our vision, is accurate and comprehensive biological diagnoses of not just AD, but TDP-43, alpha-synuclein pathology, vascular disease pathology. We want to create individualized etiologic profiles from imaging and plasma. We plan to study 2,000 individuals from the ADRCs, and there's 37 ADRCs across the country. Approximately 60% of those individuals will be impaired, 40% unimpaired with risk factors. And we plan to study them 2 time points and continue to at 2 time points, 2 to 3 years apart, and follow them over time. We're embracing the heterogeneity that the ADRCs can provide us. These centers are focusing on not just Alzheimer's, but Lewy body disease, frontotemporal diseases, limbic predominant age-related TDP-43 encephalopathy, and many other diseases. Some of the investigators in this very large program are listed here. The multi-PIs are here at the top, and some of our co-investigators leading various cores are listed at the bottom here. Well, why do we want to focus on the mixed causes of dementia? It's basically what I just mentioned to you, that this is the problem in our field that is impairing our ability to understand the effect of therapies, because we're likely treating people for their Alzheimer's disease who may or may not have some other pathology, and that's latent to us. We can't observe it, and therefore we don't know what the impact of copathology is on the treatment response that we're seeing. Why the ADRCs? These are the centers that they're already collecting these data, already have a system of sharing data with a national coordinating center called NAC, National Alzheimer's Coordinating Center. There's a strong brain donation culture in the ADRCs, and capability by mandate. All of these centers need a strong brain donation infrastructure. And there's about 18,000 participants in the ADRCs that are actively followed across these 37 sites. And collectively, this is an extraordinarily well-qualified workforce to diagnose and manage these participants. And it's a multi-site longitudinal observational study in its own right that we are now superimposing this imaging study on. We will have comprehensive neuropathology led by Dirk Keen. We will have comprehensive biofluid storage and eventual biomarkers led by Tatiana Farud and Jeff Daig at University of Indiana University, and also Henrik Zetterberg here at the University of Wisconsin. So the ADRCs with this new Clarity infrastructure will be integrated such that the genetic information, the cognitive information, and the eventual neuropathology information can all be integrated and made available to researchers like you all throughout the world to make discoveries on these kinds of data. So I'd like to kind of give you a little bit more context now of how we got here and why we want to study multipathology, and also how we want to characterize Alzheimer's disease. And I want to especially point out to you a way of thinking about Alzheimer's disease biomarkers and a way we can use them to estimate the age that a person's Alzheimer's pathology may have started in their brain. So this is just an example of two individuals, both with mild memory impairment, mild cognitive impairment. One of these people, the one on the left, we have an amyloid scan and a tau scan below. This person is positive for both amyloid and tau. The person on the right has a similar amount of memory impairment, but no evidence of Alzheimer's pathology. What is going on in their brain? These are the things we need to know, and the things that biomarkers can do for us. So a lot of the inspiration for the Clarity study that I just described is coming from the Wisconsin Registry for Alzheimer's Prevention. This is a cohort of late middle-aged adults who, many of them had a parent with Alzheimer's disease, 73% had a parent with AD. This is where they come from here on the map. And so this is a risk-enriched cohort for AD possibilities in their future. And indeed, we've been studying this cohort with biomarkers, including amyloid PET, just like the PET scans I just showed you. And here's what we find after about 15 years of studying this cohort. This is a spaghetti plot there on the left-hand side, and then reordering that spaghetti plot along the diagonal, if you will, along the slope. And so let's just go back to the spaghetti plot on the left. We see the folks in red who have amyloid elevations, and then we're following them over time. And what we're seeing is that, by and large, everyone who's elevated is rising in their rate of amyloid at about the same rate. The heterogeneity is the age of onset when they cross that dotted line, which is our threshold of positivity. So with that, we've been able to create some simple functions that allow us to estimate the age of onset. And this is fairly predictable and fairly orderly, such that we can even estimate an onset age of when a person became amyloid positive from a single PET scan. And this onset age varies quite widely. Although the mean age is about 62 years, we see here in that box plot that's on its side over there in the lower right, that the onset age can vary anywhere from early 40s to 85 and above. And this is an extraordinary thing to think about, that we can estimate an onset age of proteinopathy from a single PET scan. What I just showed you was data from the RAP study, and now I want to show you that this is not unique to RAP. The WashU data over there in the upper right, the Mayo Clinic study on aging, the Baltimore longitudinal study on aging, and even the heterogeneous ADNI study, the multi-site ADNI study. We are seeing that in these publications that we're able to estimate the onset age and therefore the disease duration of a person's amyloid proteinopathy from PET scans. This is really an important development in our field. It's going to enable us to talk to our patients about these biomarkers in terms of time, and also help us study risk and resilience in terms of time to symptoms. And this will be a really important tool for us. Here's an example of how you can use this as in understanding time to symptoms. We've plotted amyloid time, time with amyloid in the brain on the x-axis, and the symptoms, CDR sum of boxes, clinical dementia rating on the y-axis. And we see lots of heterogeneity here, but on average, it takes about 24 years or so to reach the mild stage of dementia on the CDR scale. There's quite a bit of heterogeneity such that many people are reaching symptoms earlier than the blue line would predict. And those folks who have more vascular disease burden in their brain Perhaps they have additional TDP pathology or alpha-syn pathology. We just don't know about that because we do not have a great way of measuring those other proteinopathies. We also see, encouragingly, we see people on the right of that blue line who have had amyloid in their head for quite a long period of time, 20, sometimes 30 years, who have not reached symptoms. We need to understand those individuals and learn from them how they are so resistant to eventually reaching symptoms. What about blood markers? We are very encouraged by the field in pTau 217, and this is a marker that is on many panels, including the Simoa HDX panel, but also the Alamar panel. These are results from the RAP study, as well as the Triad study in Canada and the Barcelona study called SPIN. And it's pTau 217 from the Simoa platform. We see that it's a very good, accurate test of a person's amyloid pathology and their tau pathology based on the PET studies that they also had. When we followed these people over time and looked at the blood samples that we've had in the freezer, going back to 2011 in the RAP study, we've been able to analyze all these, and it's plotted here as a function of amyloid time. These are people who also had a PET scan, and their amyloid time is on the x-axis at the bottom. For comparison, the cenyloid equivalent, the CL equivalent, is at the top, and the vertical dotted line, you see that reaching a cenyloid of 17. And that's right around the inflection point. The inflection for blood is probably a little bit earlier than a cenyloid of 17, probably somewhere around somewhere between 12 and 15 would be where that inflection point is. Over on the right side, we are showing the relationship between plasma pTau 217 concentrations and the various amyloid and tau PET combinations. Tau positivity in the medial temporal lobe versus neocortex versus both, amyloid positive, amyloid negative. And we're basically seeing a main effect of amyloid positivity, but there's also an effect of tau burden that can be approximated with pTau 217. We've also found that people who are elevated for pTau 217 have a higher likelihood of converting to symptoms over a period of about 6 years on average of follow-up. And on average, it's about 3.9, roughly 4 times more frequent progression to symptoms in people who are 217 positive. And in subsets of this, we've looked at people who are very high for pTau 217, and they certainly do progress much faster to symptoms. We've taken a similar approach to going back to imaging to vascular disease, and we've found that that we can fit a function similar to what we did for amyloid time. We can do this for vascular time, just using these very simple, easy to measure white matter hyperintensities. We can get an idea of the onset age and disease duration of a person's vascular disease. Now, just because we can measure it doesn't mean it's part of the Alzheimer's continuum. In fact, in many cases, a person does not have elevated white matter hyperintensities and Alzheimer's disease, but when they do have both, those people tend to decline much faster, as shown in the plot on the right side of the screen. Well, let's turn to the Alamar experience with this NULISA. You've already heard explained the technology behind this. I want to show you a very simple study we did. This is a cross-sectional study of about 239 people. About a third of them were biomarker negative. A little over a third were amyloid positive, and about 31% were amyloid positive and tau positive. And their demographic features are shown here on this slide. We looked at their blood samples that have been in the freezer. These were all individuals who had amyloid and tau, PET, as well as MRI and cognitive testing. We used the NULISA CNS 120 panel. Many of these people also had the Simoa pTau 217 assay, and we compared that. And we looked, we were comparing these and also looking to see what we could see with the NULISA 120 panel. So here's the first result. First of all, we compared it to the HDX Simoa, and we get very similar results. A correlation is very linear, 0.84 was the correlation. Now we're looking at the full panel of the NULISA CNS 120 among amyloid negative and amyloid positive individuals based on their PET status. And what we see is that pTau 217, 231, and 181 are the key indicators here, but GFAP and ApoE are also elevated. On the right side, we're looking at amyloid positives who are tau negative versus tau positive. And again, pTau 217 is the most robust analyte on this panel. Then finally, we looked at all comers of people who were MCI, not selected based on their PET status, but just on their symptom profile of having this clinical diagnosis of MCI. And here we saw something surprising. pTau 217, of course, was the top hit. That was not surprising. But the next one there is that on the panel, there's this phospho-TDP43 at serine 409. This was elevated, followed by GFAP and then pTau 231. And then there were some other markers there that were less significant, but still noteworthy, including oligomeric synuclein protein levels. So these are very intriguing. We're following these up. I want to show you our initial follow-up for TDP43. We used a cut point methodology based on 2 standard deviations from these scatter plots you're seeing there on the right. And we looked at the information we had available on them from MRI as well as their cognitive trajectories. The MRI trajectories for hippocampal volume are showing that folks who are elevated for TDP43 are exhibiting more hippocampal atrophy over time. And finally, the folks who are elevated for both TDP43 and who also have Alzheimer's disease proteinopathy in their brain are showing greater, a steeper slope, that is, of their cognitive trajectory over time. So greater cognitive decline among TDP43 elevated individuals. We've been encouraged by these preliminary findings. We're following these up with a more longitudinal study, and we're in the process of running those now over the next few months. So we hope to have an update on this in the very near future. But we're encouraged by this, and we're developing a new initiative around TDP43 biomarkers that Henrik Zetterberg and Hartmut Kolb are partnering with us on. And we're really excited about the possibilities of getting these two individuals together to partner with us on TDP43 biomarkers. That's mainly what I wanted to talk to you about today, and I'll just summarize by saying that AD begins 20 or more years before the onset of dementia, and that period of time is a precious period of time where all of these things are measurable and estimable from blood biomarkers using the NULISA platform. Other age-related neurodegenerative diseases are also in play during that time period, and using the NULISA platform, we hope to be able to get more insight into those other proteinopathies. We're studying the ADRC centers because there's an amazing amount of heterogeneity, clinical heterogeneity, that we will use to our benefit to make discoveries faster by working together as a consortium of centers. And finally, we're very excited about this preliminary finding with TDP43. It is preliminary and it needs to be replicated, but I think the initial finding is quite encouraging. So let me stop there and turn it back over to our moderators.
Well, thank you so much to Doug, Maura, and Sterling for your informative presentation. We'll now go ahead and start the Q&A portion of the webinar. And if you'd like to ask a question, please do so now. just simply click on the Ask a Question box located at the bottom of your screen. All right, let's go ahead and get started. We have several questions already in from our audience, so thank you so much for submitting those. Let's start with question number 1 here. Is it possible to run matrix types other than serum, plasma, or CSF on Argo?
Yeah, I'll take that one. Yeah, we have lots of experience with other matrix types. As it said, serum, plasma, and CSF are the most common, but we've done a lot of work with dried blood spots, urine, tissue lysates, cell culture media, even non-human primate samples as well. So obviously, the detectability is going to vary depending on the matrix type, as not all those proteins are necessarily going to be expected, and they're somewhat selected around serum, plasma, and CSF. Thanks.
Awesome. Thank you so much for that. Does anyone else want to chime in on that first question before we move on?
Doug's the
I think
expert.
Doug is the expert.
Sounds good. All right, let's move on to question number 2 here. What are the main differences between the inflammation 250 panel and the AQ targeted one, and the main advantages for the latter one?
Yeah, so I'll sort of focus on the more targeted panel, which is the absolute quantitation panel. So when you run that assay, you actually get relative quantitation for all the proteins in the inflammation panel, in the larger panel. So the biggest advantage is you get that data, but then for that subset of proteins, about 156, you also get absolute quantitation, so picogram per mil or attomolar level concentrations. We focused on a subset of the most important proteins within the larger panel. We also, we set very high performance criteria for all those proteins in the absolute quantitation. So that's part of the reason that just a subset have that specific performance criteria that we require. Thanks.
Perfect. Thank you so much for that. Our next question is, have you tried the NULISA panel in a longitudinal setting, and can it provide insights on trajectories of change?
I can start
Sure,
with that one.
yeah.
Sorry,
Sorry, go ahead.
Go ahead. Okay, yeah, I'll start with that one. We have not done that here in Wisconsin with our data, but we would like to, and we're planning an experiment to look at longitudinal data from NULISA. We do anticipate it will help us understand longitudinal trajectories, especially looking at clusters of proteins that may change together, or that differentiate certain trajectories. We'd love to hear what others on the panel think about that.
Yeah, like also in our case, we didn't apply NULISA in longitudinal samples just yet, but we would be very interesting to see, because in the profile that I show in the talk, we found quite a nice prognostic aspect of some of these profiles. So it'd be nice to see if we can track them over time as well. So yeah, we'll be interested to see the data as well.
Yeah. And I think from a sort of a technical NULISA perspective, maybe I would add that having such excellent sensitivity, you can detect these proteins sort of certainly in pre-disease states and then see changes from that. So you're sort of not starting from kind of blank data, but starting from a good baseline value and then seeing how those change over time.
Awesome. Well, thank you to all who answered that. All right, let's go ahead and move on here. Any specific markers for subcortical dementias?
So maybe I can take that one. So I showed this, in our data, we compare the patients with Alzheimer dementia and people with progressive supranuclear palsy, and also people with frontotemporal dementia. Basically, if the person asking this question refers to subcortical versus cortical in terms of distribution of the pathology, what we are trying to find is definitely profiles that are specific for PSP versus, for example, Alzheimer dementia. What we found from our data is particularly that in patients with PSP, the NFL was higher than in patients with Alzheimer, while in people with Alzheimer disease, the pTau, like also Sterling showed, was the primary marker, and also the inflammatory profiles were slightly different. But yeah, like, of course, it is not just a cortical versus cortical, it's also the which types of pathology we are dealing with, which type of tau we are dealing with. So definitely we think that we can find much more differences if we have like more accurate markers for the subtype of tau, for example, between tauopathies.
Great, thank you so much. It looks like we have time for one final question here, and that is going to be, can you convert the NPQ values from NULISA into PGML values?
Yeah, that's a great question. And as I said, for our targeted, the absolute quantitation panel, you do get both the NPQ and the picogram per mil. If it's not one of our absolute quantitation panels, then it's not possible to do that conversion. We use a very specific design set of calibrators that allow you to assign those picogram per mil values that don't exist within our standard relative quantitation panels.
Okay, wonderful. Well, it looks like we are at time. So before I do my final wrap up here, is there anything from our speakers that you'd like to say? Any last comments to our audience today?
No, thanks for the opportunity.
Yeah.
Thank you so much
for joining. Yeah, great to see the data from both you, Sterling and Maura.
Thank you.
Indeed, and thank you again to all of our speakers, Doug Hinerfeld, Maura Malpetti, and Sterling Johnson, for your time today and for your important research. Now, before we go, I'd like to thank our audience for joining us today as well, and for your interesting questions. I'd also like to take a moment to thank our sponsor of this webinar, Alamar Biosciences. Now, questions that we didn't have time for today and those submitted during the on-demand period will be addressed by our speakers via the contact information provided at registration. This webcast can be viewed on demand for 1 year until March 5th, 2026, so for 1 year. And LabRoots will alert you via email when that is available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. And until next time, have a great day, everybody, and we'll see you soon.